Mấy bạn có thấy được cái slide chưa?
Rồi thấy được rồi.
Ok, vậy tôi bắt đầu nha. Thank you the previous teams for your presentations, and good morning everyone. For those of you who don't know me, my name is Ling. I'm in class medicine age 44 and group 3. This is the members in my team. So today I'm gonna bring to you a presentation about nucleic acid metabolism overview. Well, my presentation has 3 parts. First, we're going to metabolism. What is nucleic acid metabolism? And then we move into digestion. How nucleic acids are digested in our bodies. Finally, we're coming to absorptions, how nucleic acids are taken in our bodies. But first, I would like to go through the history of nucleic acids. In 1869, Friedrich Mister discovered nuclein. In 1889, his student, Richard Altman, coined this molecule of nucleic acid. Between 1885 and 1901, Albert Kossel, who was a German scientist, purified the substance and later identified the nucleobases, which are cytosine, guanine, thymine, and uracil. Then, February the 28th, 1953, Cambridge University scientists who were Watson and Crick determined the pre-modern structure of DNA. And now, what is nucleic acid? Nucleic acids are the polymers of individual nucleotide monomers. As you know before, RNA and DNA are nucleic acids. Each nucleotide is composed of 3 parts. First, a phosphoric acid. Second is a nitrogenous base, and finally is a pentyl sugar, which has 5 carbons in its molecule. If this sugar is a compound ribose, the polymer will be RNA, which means ribonucleic acid. And if this sugar is derived from ribose as deoxyribose, the polymer will be DNA, which means deoxyribonucleic acid. To clearly understand the nitrogenous base, I'm gonna tell you more about its structure and classification. Nitrogenous base has 2 types. Those are pyrimidines and purines. Pyrimidines and purines are the names of the parent compounds of 2 types of nitrogen-containing heterocyclic aromatic compounds. First, purines consist of a 6-membered and a 5-membered nitrogen-containing ring. They fuse together. Purines include adenine and guanine. Then pyrimidines have only a 6-membered nitrogen-containing ring. Pyrimidines include thymine, uracil, and cytosine. Especially, thymine is only in DNA structure and uracil is only in RNA structure. I have done the introduction part. Now I would like to turn to our important parts of today. To start, I'm gonna talk about nucleic acid metabolism. Metabolism has 2 aspects. Those are anabolism and catabolism. First, anabolism is the set of metabolic pathway that construct molecules from smaller units. It is the building up aspect of metabolism, whereas catabolism is the breaking down aspect. Anabolism is usually synonymous with biosynthesis. By many chemical reactions from pentyl sugar, nitrogenous base, and phosphoric acid, nucleotide molecules are created, as you can see here. Anabolic processes build organs and tissues. They help to maintain the genetic information through many generation cells to make human's body exist and develop. And every cell in human's body can synthesize necessary nucleic acid to exist even when the body isn't provided nutrients from the food outside. Next is catabolism. Catabolism is also the set of metabolic pathways that breaks down molecules into smaller units. It is the breaking down aspect of metabolism, whereas anabolism is the building up aspect. Metabolism is usually synonymous with bioanalysis. As nucleotides can be synthesized, they can also be degraded to waste product and excreted, or can be salvaged as nucleotide components, such as purine can be degraded to uric acid, and pyrimidine can be degraded to ammonia, which means NH3. Catabolic processes are either decomposed to release energy or used in other anabolic reactions. They help to maintain the stability for our body. The breakdown of DNA and RNA is occurring continuously in the cell, so understanding analysis can help to make clear the reason of analysis process disorder. That's all basic things about metabolism. Next, we'll go to the second part of my presentation, that is the digestion of nucleic acid in our bodies. There are three enzymes attending in this process. Those are nuclease, nucleotidase, and nucleosidase. The first enzyme is nuclease, which is secreted by pancreas. It was discovered by Smith, Wilcox, and Kelly, who were working at Johns Hopkins University in 1968, isolated and characterized the first restriction nuclease whose functioning depended on a specific DNA nucleotide sequence. A nuclease is an enzyme capable of cleaving the phosphodiester bonds between nucleotides in nucleic acids, so it is also known as phosphodiesterase. Nucleases variously affect single and double-stranded breaks in their target molecules. In living organisms, they are essential machinery for many aspects of DNA repair. Defects in certain nucleases can cause genetic instability or immunodeficiency. Nucleases are also extensively used in molecular cloning. Nucleases belong to the class of enzymes called hydrolysases. Nucleases are diverse in their functions and selectivity for certain types of nucleic acid substrates. There are many ways to classify nuclease enzyme. It can be classifying depend on active position of the enzyme on the nucleic acid molecule or depend on the substrate. And the most common way is classifying depend on the position. We have two types. Those are endonuclease and exonuclease. If you read some document, you can meet the classification depending on the substrate. I mean RNA or DNA. Then we have deoxyribonuclease and ribonuclease. The next enzyme that attends the digestion process is nucleotidase, which comes from intestine. Nucleotidase plays a catalytic role in hydrolysis process, and it converts a number of different nucleotide molecules. When nucleotidase is involved in catalyzing the hydrolysis of a nucleotide, this creates a reaction as follows. A nucleotide with water forms a nucleoside and phosphate. Within the body, nucleotidase plays an instrumental in the digestive system, facilitating digestion by breaking down nucleic acids. Nucleotidase catalyzes the hydrolysis, separating the nucleoside and the phosphate, so it's considered as a phosphatase. And now it's the last enzyme, nucleosidase, which is also from intestine. The nucleosidases are continued hydrolyzed into nitrogenous base and pentose sugar with the catalysis of nucleosidase enzyme. It's also known as a phosphorylase because it catalyzes a phosphorylation, as you can see here. Okay, to summarize this part, the final products of the digestion are phosphate ion, pentyl sugar, and nitrogenous base. These products then will be absorbed. The absorption occurs mainly in duodenum and jejunum of the small intestine. At the intestinal villus, all nucleic gases are absorbed as nitrogenous base, pentyl sugar, and phosphate ions. Membrane transport proteins will carry the products of nucleotide digestion into epithelial cells from the lumen. Some involve active transport and other involve secondary active transport. Through diffusion, these products are transported from the intestinal epithelial cells across the basolateral membrane into intestinal fluid, and then into the blood capillaries of the intestinal villus. Nucleotide digestion products are transported by the blood circulation to the liver and other tissues where they undergo further degradation. Okay, now let's take a look at this diagram. This is the summary for everything I have talked. We obviously can see that the phosphate is used in the phosphorylation or excreted. The pentose metabolism is the same to usual glucose metabolism. And the last product of nitrogenous-based metabolism is uric acid, urea, and carbon dioxide. Now I have a question for you guys. So, which products in this diagram will be reused? That's my question, please. Hello?
Hello.
Hello?
Can you repeat the questions?
Okay, my question is, which products in this diagram will be reused?
The nitrogenous base, and the pentose sugar.
Anything else?
And yeah, thanks. That's all.
Thank you for your answer. And my exact answer is phosphate, nitrogenous base, and pentyl sugar. Anyway, thank you. These products that can be reused is phosphate, pentyl sugar, and nitrogenous base. They are reused to synthesize new nucleic acids or glucoside, lipid, or protein. Here. So that's everything in my presentation about nucleic acids metabolism overview. Thank you all for your listening, and now we'll go to meet the next team. Thank you again.
Thank you.
Thank you.
Xong rồi đó mấy bạn.
That's good.
Cảm ơn mọi người.
Hello mọi người.
Wow, hello vậy.
Hello Quyên.
Quyên.
Mọi người có nghe không vậy?
Không nghe tôi trả lời.
Nghe. Good morning, everybody. I'm Ring from Last YH, and today I will present to you about acid nucleic metabolism. Here is my team. My team has 16 members. My presentation includes 3 parts: 1, 2, and 3. And we move to the part 1 overview. So that you can know furthermore, I will show you a short video. Wait a second.
In today's anatomy and physiology lesson, we will be talking about how nucleus happen. Enjoy.
My DNA and RNA sensitivity.
Okay, that's the overview of digestion and utilization of nucleic acids. We continue. Okay, nucleic acids are nutritive value mainly because of the sugars they contain, namely ribose and deoxyribose. In the small intestine, pancreatic nucleus, DNA and RNAs break down nucleic acid to nucleotides, which are dephosphorilated by alkaline phosphatase to nucleosides. Later are taken up by sodium carbonyl active transport. There are 2 intestinal transporters that refer purine and pyrimidine. And now we will move to the part 2, purine degradation, and I will show you some steps involved in purine catabolism. The purine molecules AMB and TMB are dephosphorylated into their corresponding nucleotides, such as adenosine and guanosine. This reaction is catalyzed by the enzyme nucleotidase. Next, guanosine is converted into guanine by removing the ribose. Then it will be deaminated into xanthine by the enzyme guanine deaminase, while adenosine will be deaminated into inosine, then enzyme nucleosidase will hydrolyze it into hypoxanthine cytophrome. Oxidate reaction will change from hypoxanthine into xanthine by remove one F2O2 molecule. The xanthine is converted into uric acid. This reaction is catalyzed by xanthine oxidase. And the end product of purine degradation in humans is uric acid. So, how is normal blood level of uric acid? Females from 2 to 5 mg per deciliter and male from 3 to 7 mg per deciliter. The daily excretion varies from 500 to 700 mg. Nucleic acid contains in more in non-vegetarian diet. Uric acid is very solute in water. Most animals, such as ramas, birds, several insects, oxidize uric acid by the enzyme uricase to allantoin. The allantoin is then converted into allantoate and excreted in some fishes. Further degradation of allantoids may occur to reduce urea in amphibians and to ammonia in marine invertebrates. So, if you have more than 7 mg per deciliter, you will have high uric acid, and it relates to some cases such as gout arthritis, type 2 diabetes, tumor lysis syndrome, Lesch-Nyhan syndrome, cardiovascular disease, and the last one, uric acid stone formation. The first one I will tell you about gout arthritis. I also give you a short video. Wait a second.
Gout is an arthritic disease of your joints caused by too much uric acid in your body. Normally, your liver processes various substances in your body, including purines, which are components of genetic material found both inside your cells and in some foods you eat. When your liver breaks down purines, a waste product called uric acid is produced. Your body disposes of this waste through your kidneys, which remove it from your blood. When your liver produces too much uric acid, or your kidneys do not remove enough of it from your body, excess uric acid accumulates in your blood, resulting in a condition called hyperuricemia. Caffeinated beverages affect the waste removal function of your kidneys by increasing your urine output, which eliminates too much water from your body, a condition called dehydration. Dehydration can result in a higher concentration of uric acid in your body's fluids. When the concentration of uric acid becomes too high from any of these sources, it can no longer stay dissolved. The excess uric acid begins to form needle-like crystals inside your joints, most commonly in your big toe. Formation of uric acid crystals leads to an inflammatory response. White blood cells from your immune system attack the uric acid crystals, resulting in joint pain and inflammation. This is called an acute gout attack. If left untreated, elevated uric acid levels may result in lumpy deposits of uric acid around your joint, called a tophus, causing additional joint pain and disfigurement. Treatment for acute.
That is overview about gout. So we continue. Hyperuricemia with induced gout have various potential origins. 1. High fructose corn syrup and table sugar. 2. Video aggression by the kidneys. 3. Fasting or rapid weight loss. 4. Certain drugs such as thiazide, diuretics, and the last, tumor lysis syndrome. So, how many types of gout arthritis? There are 5 types. Asymptomatic hyperuricemia, interval or intercritical gout, chronic to facial gout, septal gout, and the last one, acute gout. And we have some treatment. We can use non-steroidal anti-inflammatory drugs or corticosteroids. And we move to the type 2 diabetes. Hyperuricemia may be a consequence of insulin resistance in diabetes. High serum uric acid was associated with higher risk of type 2 diabetes and metabolic syndrome, including in children. 3 is tumor lysis syndrome, an emergency condition that may result from blood cancers, produce high uric acid levels in blood. Tumor lysis syndrome may lead to acute kidney injury. Treatment includes hyperhydration. 4E Lesch-Nyhan syndrome is a rare inherited disorder, spasticity, involuntary movement, and cognitive retardation, as well as gout. Next is cardiovascular disease. Hyperuricemia may increase risk factors for cardiovascular disease. And the last one is uric acid storm formation. So, how about low uric acid? When you have less than 2 mg per deciliter, and it's related to some cases such as hyperuricemia and multiple sclerosis. And we move to the part 3, pyrimidine degradation. Okay, pyrimidine is one of two classes of heterocyclic nitrogenous base found in the nucleic acids DNA and RNA. In DNA, the pyrimidines are cytosine and thymine. In RNA, uracil relays thymine. And now I will show you some steps of pyrimidine catabolism. Step 1. Nucleotide to nucleoside. CMB, UMB, and the other side, IMB, are converted into citidine, uridine, deoxythymidine. This reaction is catalyzed by the enzyme nucleotidase. Step 2. Gemination. Citidine is geminated into uridine by citidine geminase. Step 3. Phosphorylation. Uridine are converted into uracil. Release ribo 1 phosphate by the enzyme uridine phosphorylase. By the same way, deoxy ribo 1 phosphate and thymidine are products of deoxy-thymidine phosphorylation. Step 4. Dehydration. Uracil and thymine are converted into dehydrouracil and dehydrothymine by dehydrouracil dehydrogenase. In this reaction, one NADPH plus H plus is oxidated into NADP plus. Step 5. Cyclization. Dehydrouracil and dehydrothymine are converted into beta-uridopropionate and beta-uridoisobutyrate by the enzyme pyrimidine hydrolase. In this reaction, the cyclase molecule is converted into glycerol by leaving the covalent bond at a particular place. So, step 6. Gemination. Beta-uridopropionate and beta-uridoisobutyrate are converted into beta-alanine and beta-aminoisobutyrate by the enzyme beta-uridopropionate. The secondary products of this reaction are ammonium ion and carbon dioxide, and the beta-alanine and the beta-aminoisobutyrate. These are highly water-soluble compounds. They may be extracted with the urine, or it may be transmitted to malonyl semialdehyde and converted into acetyl-CoA by transamination and hydration process. Beta-alanine and beta-aminoisobutyrate converted into malonyl semialdehyde and methylmalonyl by transamination process, and it will convert into malonyl CoA by hydration process. In transamination, it will used by aminotransferase enzyme, and the second substrate is alpha-ketoglutarate. The secondary product is glutamate. To hydration, these two products are entering the part of fatty acid catabolism like methylmalonyl pathway. So I will tell you about some clinic application. The first one is DBD deficiency. This is an autosomal recessive metabolic disorder in which the absence or significantly decreased activity of the dihydropyrimidine dehydrogenase (DPD). So that means people with this deficiency cannot break down the nucleotides uracil and thymidine. In infants with severe DPD deficiency, the signs and symptoms may include hypertonia, seizure, autism, microcephaly, intellectual disability, and the last, delayed motor skill. Individuals with this condition may develop blind retinal toxicity following exposure to 5-fluorouracil 5-FU, a chemotherapy that is used in treatment of cancer. Because the drugs are not broken down efficiently by people with DPD deficiency and build up to toxic levels in the body. And the last one is beta-uridylropionase deficiency. Loss of this enzyme function reduces the production of beta-aminoisobutyrate. It may impair the function of these molecules in the nervous system, leading to neurological problems such as developmental delay, seizures, and autism. So that's all. Thanks for watching so much. Goodbye and have a nice day.
Thank you.
Thank you.
Before I start presenting about my topic, I have a question for all of you. This is three scientists who awarded the 2015 Nobel Prize in Chemistry, and the question is, do you know the Nobel Prize 2015 in Chemistry was awarded for what? You can comment on the box or open micro to answer. Any answer? Okay, thank you. That's the right answer. The answer is mechanism of preparing DNA, and this mechanism plays role in study how cancer treatment. But we are just students, so before understand the complexible mechanism, we must find out some basic knowledge about DNA or nucleic acid. And that's the topic I am going to talk today. I will present about DNA replication, transcriptions, and medical application. The first one is DNA replication. For simple, DNA replication is the process producing two identical replicates from one original DNA molecule. This process occurs in all living organisms and is basic for biological inheritance. And the question is, when will DNA replication occur? You can see interface here. This is the longest period of the cell cycle, and DNA replication occurs in the phase of interphase. Next is the enzyme. You may find the name of the enzyme are very familiar we have non-H in the hyphens. This enzyme will catalyze various steps in this process. For example, DNA ligase will unwind and requires DNA chains to prevent the DNA from becoming subcoiled. Besides the enzyme, we also have some components are needed, such as SSB proteins binds and prevents DNA double helix recoil after DNA replicate in Y cells. Remember these enzymes, and I will talk more particularly about their function when going into the process. The rules there are two main principles to keep in mind are replications is a semi-conservative process. This means a new DNA consists of one change from mother and the other is complementary from environment. The second is specificity of base pairing. This means adenines will connect to thymine by two hydrogen bonds, and guanines will connect to cytosine by three hydrogen bonds. Now is the most important thing. DNA replication has three stages: initiation, elongation, and termination. And I will resend through this video. The first step is separate the two chains. Enzyme unzip a helicase. It makes a replication fork. After separate, each chain will be a template for creating a new chain of DNA, and DNA ramase will generate RNA rimers. Rimers are short RNA molecules that mark a starting point of DNA replication. After that, DNA polymerase trees bind to the rimer and make new chains follow complementary principle. And this enzyme has only one direction, 5' to 3'. This is very important. It is one reason that we have two kinds of chains. Leading chains is made continuously and lagging chains is run in opposite direction. Therefore, it's only made from series of small fragments called Okazaki. Next, in elongations, the rimers are formed by DNA ramase and DNA polymerase tree continue each other, and this process will repeat again and again in other positions. Once new chains is made, exonuclease will remove or write RNA rimers from both chains of DNA, and DNA polymerase one fill this gap. Finally, DNA ligase will connect fragments to form a continuous double chain. That's all about DNA replication. The second part is transcription. Transcription is the synthesis of RNA molecule by using a DNA molecule as a template. It transfers the genetic information from DNA to RNA. And when this process occurs, its difference in eukaryotic cell it occurs in nucleoid regions, and in eukaryotic cell it occurs in nucleus. Transcription also has three stages. In initiation stage, RNA polymerase bind to special regions in DNA called promoters. In humans, promoter is GA box. This unwinds the DNA double helix and provides single chain template in 3 fragments to 5 fragments need for transcription. Transcription starts from here in the initiator site. In elongation state, RNA polymerase move along the template DNA chains in 3 fragments to 5 fragments by an mRNA out of the complementary space pair. You can see messenger RNA is have direction 5 fragments to 3 fragments. And finally, the termination. Once the DNA polymerase bind to sequence called terminators, the transcription stops. The RNA molecule and RNA polymerase are released. Which region in genes is transcript already will rewrite immediately. Now there are some differences in progressing mRNA we have to notice. In eukaryote, the RNA transcripts are ready to be used directly as a template for translation. And in eukaryote, the RNA transcripts are called re-mRNA and must go through extra resin called RNA lysing before it can direct translation. For particular process, look at this video. In eukaryote, the mRNA has many intron and exon. The mRNA will be added of 5' caps at the beginning and the 3' poly tail at the end. Then lysosome will remove intron and exon are stuck back together to make the major mRNA that leave the nucleus enter the cytoplasm and translation begin. You can see the cap and the tail are rotating mRNA when it goes through nucleus spore. Now we move to transcription factors. Transcription factors are proteins that regulate the transcription of DNA into RNA. They control the beginning of transcription. RNA polymerase can attach to the promoters only with the help of them. Some transcription factors called activators that activate transcription. They may help the basal and RNA polymerase bind to the promoter. Other transcription, reverse transcription, they are regressor. The regression can work in a variety of ways. So these important proteins can help determine which genes are active and in each cell of your body. In addition, some group of transcription factors binding sites called enhancer and silencer can turn a gene on or off in specific spots of the body. Next is RNA polymerase. Is the enzyme that is responsible for copying DNA sequence into RNA sequence. Like DNA polymerase, RNA polymerase catalyze the formation of ester bonds between nucleotides that pair with the complementary nucleotides on DNA template. But unlike DNA polymerase, RNA polymerase initiate the synthesis of new chains in the absence of primer. And final problem I want to talk is type of RNA polymerase. Difference between RNA polymerase of eukaryote and eukaryote. It is summarized in this table. And you can see in the other table, RNA polymerase also has 3 types, like DNA polymerase. Next part is medical applications and book will be sent to you.
Hello everyone. So following the presentation, I will talk about how we use this knowledge to apply in clinical. First, we move to applying in diagnosis. There are two things that I think really important. That is PCR and recombinant DNA. First, PCR. PCR is stands for polymerase chain reaction. It can detect rapidly and accurately the residence of bacteria such as chlamydia, mycoplasma, mycobacteria, herpesvirus, and enterovirus directly from clinical specimen. Its components are nucleotides, ramer, tap DNA polymerase, reverse transcriptase, and thermocycle procedure. PCR involves 4 steps. First, we need to extract nucleic acid from the microorganism. Next, we will naturation at 90 to 95 Celsius degree. We will separate the extract double strand DNA into single strand. The third step, that is annealing, we cool into 55 Celsius degree. We cool into a low rimer, as you can see the black one here, to bind to the single strand that we separate. And the last step, that is extension. The enzyme Taq polymerase and nucleotides are added to create new DNA fragments complementary to the target DNA. And after that, we will repeat a lot of cycles, and the result is an accumulation of specific target sequence. As you can see here, a lot of target sequence. That's PCR. Next, we move to recombinant DNA. But first, before we go into detail, I need you know two fundamental difference between PCR and recombinant DNA. Recombinant DNA is the replication of the DNA within a living cell, and it's cutting and pasting DNA sequence. In contrast, PCR is the replication DNA in the test tube, which is free of living cells, and it is amplified by copying an existing sequence. Recombinant DNA has 7 steps. First, we need to choose the host organism. As you can see here, the host cell. Next, we will repairing a vector, repairing a DNA. Next, we will cutting vector and DNA by restriction enzyme. Then we will combine it together and create the recombinant DNA. That is the step 4. Step 5, we will introduce it into the host cell. Next, we will selection of organism that containing the recombinant DNA. And the last study, we will screening for lungs with desired DNA insert and biological properties. So how we use recombinant DNA in our career in the future? Recombinant DNA is used to reduce human insulin, human GH, and blood protein factor 8. We also can use this to diagnose genetic disease, then diagnose microorganism infection like HIV, or maybe diagnose in tumor pathology like leukemia, lymphomas. Yeah, and that's all about diagnosis. We move into treatment. The first target in treatment is hepatitis B virus. This is the structure of the virus that we have learned in microorganism. This virus will affect the liver cells, as you can see here, and it will through a lot of steps to generate their to create their new generation and also damage the liver cell. So if we use drugs that terminate those of this virus step, this virus will not harmful with our body. So that is the work of nucleotide analogues. How it works? it will fool the hepatitis B virus into thinking that they are normal building blocks for DNA. Next, we will, we will use this to treat cancer. How they work. These agents behave as anti-metabolites, compete with physiological nucleosides, and interact with a large number of intracellular targets to induce cytotoxicity. We have some example in here. Pyrazolopyrimidine or allopurinol for short is used for treat high uric acid in blood or gout because it inhibits synthesis purine and xanthine oxidase action. Nucleotides contain arabinols, which is used for cancer treatment and viral infection. 5-iodo-2-deoxyuridine is used for keratitis by herpes. And the last one, that is 5-fluorouracil, is used for cancer treatment. Next, we go to nucleotide excision repair. There are three excision repair pathways exist to repair single-strand DNA damage. That is nucleotide excision repair, base excision repair, and DNA mismatch repair. So DNA damage occurs constantly because of chemical, because of radiation, and also because of UV light, ultraviolet. So with this technique, we can recognize the damage by UV lit, and then they will remove the short single-strand DNA segment that contain a lesson. So as you can see here, this is the lesson which is created by UV light, and after a lot of sequence, it will remove this lesson, and the undamaged single-strand DNA. As you can see here, we use DNA polymerase to synthesize the new shortened complementary DNA. The last one is gout. So what is gout? Gout is a systemic disease that results from the deposition of monosodium urate crystal in tissue. So why there are deposition? There are two causes. Maybe that they are over radiation, or maybe they are under expression. With under expression share a major percentage, about 90%. Over radiation, this come from dietary purine from your food like meat, seafood, or beer. Endogenous purine synthesis as malignancy, tumor lysis syndrome, which is mentioned before. Purine civet, that's HGPRT deficiency. This is an enzyme in the purine civet, and when it deficiency, it causes hyperuricemia. And the last one is purine regna. You can see in glycogen storage disease. In under expression may come from you use diuretics, maybe your kidney has some problem in renal failure, or maybe you consume a lot of alcohol, you have a genetic defect. All of this, and with this risk factor like male, age, and obesity, will create hyperuricemia, and it will increase the renal and gastrointestinal tract excretion. So how we treat it? We will use allopurinol and oxypurinol. Both inhibit xanthine oxidase, an enzyme in the purine catabolism pathway that converts hypoxanthine to xanthine and to uric acid. Adverse effects: it will increase incidence of acute gouty flares, maculopapular pruritic cracks, maybe gastrointestinal adverse effect, including nausea and diarrhea. So that's all. This is the member list of our team, and this is the reference that we use for this PowerPoint. Thank you for your listening and hope you enjoy it.
Thank you.
Thank you.
Alo, mọi người có ai rõ không vậy?
Có.
Okay. Okay, hello everyone. Here we come to the presentations of group 4 about specific metabolism of nucleic acids. And our instructor is Hà Thị Thảo Mai. These are our members. Today we have 6 parts total. And the first one is uric acid metabolic disorder. So we should find out the definitions of uric acid. So uric acid is the final product in the process of degradation of purine in plant, red meat, seafood, and alcoholic beverage. The enzyme deficiency is hypoxanthine-guanine phosphoride oxide transferase, we can call XPRT for short. It's one of the central enzymes that make nucleotide from base purine. And look at this image here. You can see that XPRT is very important to synthesize nucleotides. If XPRT decreases, it will become to uric acid. And the consumption of high purine food will make the kidney cannot remove uric acid. And when uric crystals are deposited too much in the kidneys, forming kidney stone, glomerular fibrosis, interstitial nephritis, especially this overproduction of uric acid we can call hyperuricemia, leading to gout. And gout patients have hyperuricemia in the synovial fluid, leads to the formations of uric crystal precipitate. And when the cartilage is damaged, it will attack to the bone below and leading to trophy cyst formations. And the final is will destroy the bone. And the second part today is adenosine deaminase, is standard for ADA. ADA catalyzes the amination of adenosine to inosine. Here. About specific features, ADA is produced by cells throughout the body and is associated with the activations of lymphocytes. For example, Mycobacterium tuberculosis cause increase the amount of ADA. An ADA test measures the amount of adenosine, the aminase present in pleural fluid, so the doctor can diagnose a tuberculosis infection of the pleural. We must be patient that ADA test is not a diagnostic test and does not prepare for the cultures of diagnosing tuberculosis because it may be positive even when the number of bacteria are very low. The test is recommended in some cases, like migrated from blood spot, pregnant people, be in close contact with someone who is suffering from tuberculosis or have a very low immune system, children who have positive in the tuberculosis screening test, who in the hospital are social worker also. Okay, let's move to DNA nanotechnologies. DNA nanotechnologies is the design and manufacture of artificial nucleic acid structures for technological use. Nucleic acids are used as non-biological engineering materials for nanotechnologies rather than the carrier of genes, and forming DNA to two-dimensional and three-dimensional structures for medical purpose through our four structures below. And one of these DNA structures, DNA's origamis, is the nanoscale, folding up DNA to create two-dimensional and three-dimensional shapes. The long single-stranded DNA is folded into the desired shape by multiple short single-stranded DNA. So we can realize that DNA is used as scaffolding for holding other molecules in place or to create structures. And about its applications, the first one is pathology resolution to improve controls of neuron prosthetic and prosthetic limbs, or smooth the irregular neuroelectrical activities, or amplify neuronal signaling. The second application is making smart drugs for target drug deliveries, as well as for diagnostic applications. This image is about nano capsule, and this one is about treatment of respiratories. And this one is about treatments of pulmonary fibrosis. And the final applications is environmental support in neuronal repair and activate neuronal array. Okay, let's talk about genetic engineering. Genetic engineering is the direct manipulation of an organism genes using biotechnology to change the genetic structure to make biological products or better organisms by using recombinant DNA methods or by artificially synthesizing the DNA to change the genetic material. And genetic engineering have numerous fields in fixing severe genetic disorder in humans, just vaccines and other products from organisms for crop and in crop for nutritional values and tolerance to environmental stress. Okay, we go to recombinant DNA. Recombinant DNA technologies is the turning of DNA molecule from two different pieces here. And here are the products. The recombined DNA molecules is inserted into a host organism. And in this image, the host organism is bacterial cells to produce more and new genetic combinations. And about applications is to identify, map, and sequence genes and to determine their functions, or analyzing gene expressions, or examining protein synthesis in cells and organisms. And other applications recombinant proteins is to modify gene sequence and useful products in human insulin, growth hormone, or blood clotting factor 8, hepatitis B vaccine, etc. Okay, we go to DNA fingerprinting. And as you know before, this is what makes DNA fingerprinting as a useful tool. It allows us to identify an individual from biological samples. It's called fingerprinting. And the application is physically connect a piece of evidence to a person or rule out someone as a suspect, show who your relative, or identify a dead body that is too old or damaged, match tissues of organ donor with transplanted people. And as you can see, this have total of 6 steps. Step 1 is extract DNA nuclei from the cells. Step 2, they use PCR for amplifying the DNA. Step 3 is to cut DNA strand into fragments in specific positions by restrictions enzyme. Step 4 is sorting the the DNA segment by Chell's electrophoresis. Because of the negative charged molecules, the shorter segment the farther is moved away our original locations. And step 5 is loading the DNA gel onto a suitable membrane. Single-stranded DNA fragments are strengthened on the membrane. This hybridized with a suitable DNA broth. The DNA to be investigated like DNA found at the prime site. And before these broths are used, they are tagged with a radioactive substance or fluorescent dye. And the final step, we can see the result by autoradiograph. And this is a technique using X-ray film to visualize molecules or fragments of molecules that have been radioactively labeled. And the final part today is SNRNBS and human disease. SNRNBS is a class of small RNA molecules found in nucleus of eukaryotic cells, approximately 150 nucleotides, and the most common human snRNA component is included from U1 to U6, except for U3, by also RNA. And the goal is to transform the mRNA here into a mature and fully functioning mRNA by cutting the intron and splicing the exon together after that. Okay, about human disease. The consequence of spinal muscular atrophy is muscle weakness, poor muscle tone, and the causes is the mutations in the survival motor neuron 1. This keratosis congenita is the mutations in the assemblage SNRNBS. This disease will express in skin, nails, and mucous membranes. The last one is Prader-Willi syndrome. Prader-Willi syndrome will make the patient feel extreme hunger, confusion, and behavior problems related to the of our presentations. Thanks for your listening. And do you have any questions?
Không ạ.
Okay, goodbye. Thank you very much.
Thank you.
Thank you.